Question number 96. The common features between Cara and Marchantia are A. Dioecious, B. Flagellated antherozoids, C. Jacketed sex organs, D. Protogynous, E. Female sex organ is nucule or oogonium. First option only A, B and C, second only B and C, third only A and E, fourth only B, C and D. So you have to choose the common features between Cara and Marchantia. But before solving this question, you should know what is Cara and Marchantia. Cara is a green alga, whereas the Marchantia is a liverwort. It's a bryophyte. Now, the common features. A. Dioecious. Dioecious means when both the sex organs are present on separate plants. It is called dioecious condition. In case of Marchantia, it is true. Marchantia is dioecious as it has male and female sex organs on the separate plants. But it is not true for Cara because most of the species of Cara are monoecious. So this cannot be a common feature between Cara and Marchantia. So this is ruled out. B. Flagellated antherozoids. It is true because Cara and Marchantia both have flagellated male gametes or flagellated antherozoids. C. Jacketed sex organs. Although the algae have non-jacketed sex organs, but the exception is Cara. Cara bears jacketed sex organs like Marchantia. So this is also a common feature between Cara and Marchantia. G. Protogynous. What is protogynous condition? Protogynous condition is the condition when a female sex organ matures first before the male sex organ. But this is not true for either of them because Cara shows protandrous condition where male sex organ matures first as compared to the female, and in Marchantia both the sex organs mature at the same time. E. Female sex organ is nucule or oogonium. This is true for Cara but not for Marchantia. Why? Because in Cara the female sex organ is called nucule or oogonium, but in Marchantia the female sex organ is called archegonium, not the oogonium. So this is not the similarity. So similarities are B and C only. So the correct option for the given question is option number 2. Let's move on to the next question. Question number 97. Mark the odd one with respect to the post-fertilization events occurring in a flowering plant. First option, embryo formation. Second, ovule develops into a seed. Third, gametogenesis. Fourth, ovary ripes into a fruit. What are post-fertilization events? The events which take place after fertilizations. They are called post-fertilization events in a flowering plant. Embryo formation. Embryo forms from a zygote, and zygote is a result of fertilization. So it's a post-fertilization event. So this is ruled out as we have to choose the odd with respect to the post-fertilization event. Second, ovule develops into a seed. It is also a post-fertilization event because after fertilization the ovule develops into a seed. So this is also ruled out. Fourth, ovary ripes into a fruit. When ovary becomes a fruit, it is also a post-fertilization event. So this is also ruled out. Gametogenesis is formation of gametes, and gametes are formed before fertilization so that gametes can fuse to form a zygote. So this is a pre-fertilization event, not the post-fertilization event. So the correct option for the given question is option number 3. Let's move on to the next question. Question number 98. A typical angiospermic anther is A. Bilobed, B. Dithecous, C. Tetragonal, D. Tetrasporangiate. First option only A, B and D. Second, all A, B, C and D. Third, only A and D. Fourth, only B and D. So you have to tell the correct option regarding the angiospermic anther. So this is the figure you can see here. This is a TS of the tetrasporangiate anther of an angiosperm. Now you can see here it is bilobed. This is single lobe and this is another lobe. So it is bilobed. It is dithecous and this is one theca and this is the another one. and it is tetrasporangiate. Why? Because this is one sporangium and in each corner you can see a sporangium. So together they are 4 sporangia called tetrasporangiate, and it is tetragonal too. So it is bilobed, yes. Dithecous, yes. Tetragonal, yes. Tetrasporangiate, yes, as it has 4 sporangia. So all A, B, C, D are true regarding the angiospermic anther. So the correct option for the given question is option number 2. Let's move on to the next question. Question number 99. The cells of anther wall layer which nourish the developing pollen grains is related to all of the following features except first option dense cytoplasm, second polyploid, third multinucleate, fourth low DNA content. First of all, you have to identify the which layer of anther this question is talking about. So it is a hint over here that the layer which nourish the developing pollen grains. The layer which nourish the developing pollen grains in angiosperms, it is tapetum. So you have to choose the correct option regarding the tapetum. The cells of tapetum contain dense cytoplasm. This is true. They are generally multinucleated. This is also true. They are polyploid, means the copy number of chromosomes is more than 2. It is also true. Fourth one, low DNA content. No, tapetum has high DNA content due to polyploidy, endomitosis, etc. So they have high DNA content, not the low DNA content. This is not true about the tapetum. So the correct option for the given question is option number 4. Let's move on to the next question. Question number 100. If a typical angiospermic anther has 25 microspore mother cells in its each sporangium, calculate the total number of microspore tetrads, pollen grains and male gametes respectively produced by this anther. First option 25, 100, 200. Second 100, 400, 800. Third 100, 100, 400. Fourth 50, 200 and 400. This question is talking about the typical angiospermic anther which has 25 microspore mother cells in each sporangium. In the just previous question you've seen that this, if you see this TS of the typical angiospermic anther, you can see there are 4 sporangia in each anther because anther is tetrasporangiate. If each sporangium has 25 pollen mother cells or the microspores mother cells, then in total there will be 25 into 4, 100 pollen mother cells or the microspore mother cells in an anther, because in the question it is asked by this anther. So in the anther it will be 100 PMC, 25 in each sporangium. Now you can calculate easily. One pollen mother cell gives you 1 tetrad or the microspore tetrad, which gives you 4 pollen grains. And each pollen grains gives you 2 male gametes. So 4 pollen grains will give you 8 male gametes. Now multiply everything with the 100. Why? Because we have to find out from the 100 PMC. So 100 pollen mother cells will give you 100 microspore tetrads, 400 pollen grains and 800 male gametes, because you have to multiply this by 100, which gives you this, again 100, again 100 and then again 100. It will give you 800 male gametes. So number of tetrads will be 100, pollen grains 400, male gametes 800. So the correct option for the given question is option number 2. Let's move on to the next question.